Yeah. Thank you very much for the invitation. It's great. I hope we'll have a great time here. It was a very nice introduction to the Chromatin organization at the start of this session itself. I'm going to talk a slightly little bit different aspect of Chromatin organization, but more in bacteria. Okay. So I'm from IISc in biochemistry department there, joined around 4 years ago, exactly 4 years ago actually. We're working on different aspects of chromatin organization. Some part of my lab works in the chromatin organization in human cells itself. Other part more in vitro purified components. We look at how they work, what is their mechanism. Right, to start with, this is the sort of a central theme of this conference, where you have bare DNA, which is wrapped around histone octamer, gives again, then you have this local topologically associated domains. They appear via, we think, by loop extrusion process or so, and then those domains could come together depending on the kind of modifications that may there on the histone tails itself. They can be more expressed in terms of transcription, or maybe they are suppressed. Or compartment A or compartment B, they call. And then individual chromosomes, of course, they sit in the nucleus, what they call chromosomal territories. I've seen that there is a talk in after two more talks on the multi-scale organization of the genome, so I'm not going to talk about this, but my student has a poster on some part of this, how it works. Abhinav Banerjee, please go there and see that poster. So this is the aspect of more eukaryotic nuclear chromatin organization. But when it comes to bacteria, it's very different. You don't have those nucleosomes what you have seen in the previous talk, right? But you have a bunch of other proteins. Actually, around 10 or so, less than 10 different proteins they are there. They organize the bacterial chromatin. They all work in very different way. Their mechanisms are different. For example, some proteins bend the DNA, others only bridge two different filaments of the two different DNA molecules, or maybe they also do, some of them can actually make loops in the genome. That's what you see. And then there's also supercoiling on top of what you see in the human or eukaryotic chromosomes in bacteria when it comes to bacteria. So we are looking at aspects of organization, bacterial chromatin organization, by more purified proteins. We purify the proteins and then look at them at single molecule level under fluorescence microscope. Right, so this is a single chromosome copy of a E. coli bacterium done long ago by Ruth Kovanov. This was under electron microscope visualized. So single chromosome taken out of the bacteria and then washed with high salt so that you remove all those local proteins, all the nucleide associated proteins, and end up having what you see is that large loops emanating from the center and ending there itself. But you also see this intertwined DNA structures. These are supercoiled DNA. So bacteria takes advantage of both nucleide associated proteins that gives local structure, but also supercoiling so that it can condense into bacteria. If you place bacteria here, it will be somewhat size of this much here only in the very center, explodes around 100 folds or so, or you have some 1,000-fold longer DNA molecule that fits into the bacteria. All the protein components and the supercoiling take care of the compaction in bacteria. So I'll talk about what is the sequence that governs the supercoiled DNA position. One aspect, other thing, how this loop may arrive in, for example, bacteria or our own cells or so. And also I'll talk about a component that gives global genome organization, also compaction to the bacterial chromatin. Right, when it comes to supercoiling, they actually have big role in almost all the processes related to DNA, including transcription, replication, and of course, it, as I said, it condenses DNA. And also they give rise to different structures, local structures to the DNA. They may have roles, different roles themselves also, those structures. For example, supercoiling can bring in two different distant loci closer to each other that can help in gene regulation, for example. Or you can have, if you have a negative supercoiling that can open the genome, so then transcription can effectively work there, or more processively work on such DNA. So these are some of aspects that can supercoiling affect. But when we started looking into this aspect of supercoiling, we didn't know much about the dynamics of supercoils. How do they may exist on a piece of DNA if you have supercoiled DNA? Or if they exist, do they exist as a on a long piece of DNA? A single plectonymic structure, that's what you get if you twist the DNA. And if they occur, what is their dynamics? Do they sit at one position or do they diffuse, or what happens? All those aspects were absent. So that's where we developed this assay called, we call it intercalation induced supercoiling DNA assay, where we immobilize a DNA, long piece of DNA we take, is around 21 kilobases in this case. DNA is modified at the ends with biotins, and then we immobilize on the surface using biotin streptavid interactions, one of the strongest non-covalent interactions that are available. So because the DNA, only ends of the DNA are immobilized on the surface, and these interactions are via multiple biotin streptavid interactions, that gives topologically closed structure here. So that means the DNA in the center rotationally constrained. So the way we introduce supercoils into this DNA by adding an intercalating dye called cytoxorange, doesn't matter, you can use some other dye also if you want. So what happens is that when the dye binds on the DNA, locally unwinds wherever it binds. Since the DNA length is around 21 kilobases, hundreds of such molecules can bind on the DNA. Each of them can unwind a little bit. So that unwind torsional stress can accumulate into structure like this. That's what you see here. For example, each of those are individual DNA molecules immobilized on the both ends. So you're seeing one molecule here. These bright spots are these plectonic structures. What you see is that they diffuse along the length very fast. The diffusion is, of course, the torsion itself diffuses across the length so that the plectron can diffuse. And other thing is that, of course, these are plectons. We make sure that by looking, if you keep on observing, for example, one of these molecules, it will disappear at some point. It will become like molecule like this. They wiggle perpendicular length. Yeah, you see there, the bright spot disappeared. This is because of the photo-induced nick in the DNA that can release the torsion. So what we are seeing here is that supercoiled DNA, because DNA is immobilized on both the ends because of the torsional constraint, and that constraint, upon adding cytoxorange or intercalating dye, that gives you supercoiled DNA. Now we have an assay that allows you to visualize supercoiled DNA. Perhaps you can also study proteins that could interact with supercoiled DNA or maybe weigh DNA also in a single assay. So Sneha, my student, is going to talk about that aspect of how supercoiled DNA would influence nucleotide associated protein, one nucleotide associated protein. She will talk in a couple of talks later. Right, so now I'll talk a little bit about, where is the position, what position or which sequence on the DNA are more favorable for this plectonymic DNA to occur. So for that, we took two different DNA molecules, a DNA molecule of almost homogeneous GC content, other one has a little bit fluctuating GC content. You can see that there is low GC content, high GC content. Then we measured the number of plectons would form at each position over the time and make a time average. That gives us a density of plectons over the time. When we measured, we see that very different plectonium density for two different DNA sequences. That already says that the sequence actually governs the position of the plectonium, meaning that if you take a bacterial chromatin, some positions on the bacterial genome have higher propensity for formation of the plectonium than other sequences. Of course, it's not very clear if you look at GC content alone here. There is no real correlation between the GC content and the plectonium density formation. But what was more interesting is that when we looked at the way nucleotides were arranged, we did small calculations on what is the local curvature of the DNA. When we looked at the local curvature, we could see that locally curved nucleotide sequences would pin the plectonium with more probability than other sequences. So for example, here you see this higher plectonium density at this piece, right? That's the one which has a higher local curvature. We did multiple experiments to prove that it is actually the local curvature. And we also went on to check where are those local curved sequences there in the genome bacterial, for example, E. coli bacteria. We found that upstream of the promoter, 100 bases upstream of the promoter, there is always a curved sequence. Of course, the curvature varies a lot depending on the promoter, but they are there, and they do pin the plectoniums when we plugged in such sequences in our assay. For example, here in the center, we see a plectonium density would appear. It says that plectonium would pin right before the promoter site, but we don't know for what reason, whether that will contribute for the transcription or not. That could be something we will be looking in the future. Right, our model is this, that if you have a straight DNA molecule to bend into a plectonium, you have to spend that much energy. But if you have already, locally curved sequence, you spend much less energy. Of course, then because of the pre-curvature in the DNA, the plectoniums would pin there with more probability. So indeed, we actually made a DNA based on this model where you would have a very high propensity for plectonium formation based on this physical model. And then we could actually, when we measured, we see a very good correspondence between the model and the data. Right. So here, from this part, I would say that we sort of understood that the genome sequence or the bacterial DNA sequence not only encodes for the proteins, but also encodes for its supercoiled DNA structure. So thereby it regulates probably the the DNA transactions, we think. Right. The other aspect of the bacterial genome, or maybe also any other genome, if we think that there are large loops, right? I'll talk a little bit quickly about that, how those loops appear. For example, in this case, we took example protein SMC condensin molecule, which the previous speaker introduced a little bit. So we purified this entire complex, and then, so the complex, if you remove the complex from the cells, of course, the cells have a hard time, they can't propagate any further. That means condensin is very important. So we purified the protein and assayed on the similar assay where the DNA is again immobilized on both ends, but this time with single wide streptavidin interaction so that you don't have torsional constraint on the DNA. So you can see the DNA nicely. When you stain without the protein, you see almost homogeneous intensity across the length. But as soon as we add condensin and ATP, the energy that condensin requires, we see that the bright spot appears in the center or somewhere along the length. And those bright spots are because of a active loop extrusion process. You see that DNA is immobilized here and here. There's a bright buffer flow there. So a bright spot appears here, and then the spot size keep on increasing over the time by pulling this entire DNA length into this loop. So in this case, it's condensin molecule from yeast. What it does, it's a single-sided loop extrusion. It's pulling only from DNA from one side. This is a real-time movie. You can see the time scale there. We can indeed quantify how fast it loop extrudes. For example, here you see in a couple of seconds or 10 seconds or 20 seconds or so, around 30 kilobases DNA being pulled into loop. Roughly it comes to around 1 to 1.5 kilobases per second when it comes to this one. So this was back in 2018, was the first time we were showing loop extrusion and single molecule experiments. And this is the condensin from yeast. But now there are a bunch of other SMC molecules, coercin, human condensin 1,2, SMC 5,6 from humans itself, or bacterial SMC. All of them are shown to have perform very similar activity using the exact same assay. I'll switch gears to something else that we recently started looking into, the mechanism of DNA compaction by naps in bacteria. This is unpublished data here. So again, as coming back to this picture, what I was showing that, you have a bunch of different proteins. Each of them have a different way of interacting with DNA. Some of them sharply bend, or some of them bend less, or they stiffen the DNA, or they can bridge the DNA, depending on the kind of molecule you're looking at. So we purified the nucleotide-associated protein from Mycobacterium tuberculosis, and then we looked at that one. So we know that Mycobacterium tuberculosis, of course, is a big problem. Almost 1/3 of the human carries this bacteria in latent phase, of course, but then anytime it can activate. So it's good to learn how this bacteria itself survive in the host, inside the host, so that we may find ways to disrupt their mechanism of the way they work there inside. So previous work showed that LSR2, one of the protein from bacteria, this is a nucleotide-associated protein we are looking at. The protein is very important, apparently. So when you delete that nucleotide-associated protein LSR2, they could see that the disease pathology is reduced dramatically. This is in mouse lungs. You can see that. And what we also know from previous studies that LSR2 binds on AT-rich sequences, meaning that each protein might have preference for slightly higher AT-rich sequence. It also regulates large number of genes, or around 21% of the genome is being bound or compacted by LSR2. And it is, so based on this study, we know that it is important for its survival and pathology of the bacteria. Right, so here is the structure of the protein. Maybe it will look interesting if I explain a little bit more about what is the structure. So for example, here, this is the DNA binding site of the protein, and this is the dimerization domain, so it can dimerize. When it dimerizes, of course, you will end up having two different binding domains, and these two domains are connected via intrinsically disordered region full of lysine signed arginines, RGGs motifs, which are known to form phase-separated condensates. And it also has a tail at the C-terminus. Once it dimerizes, upon binding to DNA, people speculated that this tail of the C-terminus could form C-terminus interactions with other proteins so that it can oligomerize. Once it oligomerizes, of course, you have more and more interactions or more binding sites on the DNA that facilitates more ways of interacting with DNA. This sounds like a very good recipe for formation of phase-separated condensates, especially when it binds on the DNA, having multiple binding sites on the DNA and also the intrinsically disordered region. So yeah, this is the schematic, the way we show that it can dimerize in, these are two different dimers shown in different colors. It can dimerize via the folded region in the center. It can also, it has this disordered region, and then C-terminal DNA binding domain is shown in blue color, and then can also oligomerize via C-terminal interactions. Right, so this flexible linker facilitates to bind between different regions of the DNA. We think that actually helps in forming the phase separated or form to undergo phase separation in DNA. So we purified the protein. We could see that it actually nicely compacts, in a concentration dependent manner, binds on the DNA and compacts. Then we also seen that it can form this bridging complex of DNA. By the way, this is nothing new. People have seen this, that they actually proposed that LSRT is a DNA bridging molecule. But what we wanted to see, how does it form at a specific locus or specific loci in the DNA? That's what we want to see. For that, we purified the protein, and then we could see that, as I said, the protein has the architecture that can actually be a perfect candidate to study phase separation because of the multimerization and also intrinsically disordered regions. They are not so surprisingly that we could actually see that protein undergo liquid-liquid phase separation itself at moderately high concentrations. But when we when we added the DNA to protein, we could see that it actually phase separated much lower concentrations, probably much lower than physiological concentrations where protein exists. And then, obviously you can see that they are spherical in shape. That sort of tells that they are more like liquid-like nature. But to prove that, to see into the liquidity of the condensates, we did FRAP, where you photo bleach the center of the condensate. You can see that it recovers within 10 to 15 seconds or so. So that means it's really high fluid. It is fluid in nature, these condensates. Right, to understand which of these structural motifs or the DNA protein motifs that play in role, we looked at each of the components. For example, dimerization domain. We could introduce mutations in this domain that will basically kill the dimerization. We wanted to see whether dimerization is important or not for the phase separation. Those are the two different mutations we introduced and seen. Other thing is that the oligomerization domain, the C-terminus, once it dimerizes, it can also oligomerize through C-terminus. That can leads to oligomerization. So we wanted to check oligomerization is important or not. So we generated a truncate of the protein that doesn't carry any more N-terminus. The other part, we also removed the IDR. We did several mutations in the intrinsically disordered region, including converting some of the lysines to alanines, arginines to alanines, or we also truncated the entire IDR to see how each of those parts of the protein, each of those structural motifs play a role in liquid-liquid phase separation. So here I'm putting all the data one go that, for example, when we introduce the mutation, the dimerization, this mutation kills the dimerization. We don't see any more phase separation as we see in the wild type. When we deleted the IDR region, also again we don't see much any phase separation that what we have seen. Also, when we mutated some of the arginines, 3 arginines in the center of this IDR region of around 20 amino acids, that already reduced somewhat the phase separation. So this basic data says that both dimerization and oligomerization and also the intrinsically disordered regions, all of them are important for the protein to function and form phase separated condensates. The next thing we did was single molecule experiments to understand how this protein as a single protein can regulate large number of genes. Around 20% of the genes they regulate in the bacteria, right? So we wanted to understand how it can specifically bind on each of those genes. So the assay was again very simple, as I showed previously, that you have DNA, we have DNA immobilized on both the ends, then we add the protein in this case to see what happens. So initially the DNA is visible here because intercalating dye binds all over, but over the time, once we introduce the protein here, you see the protein start to form a local cluster here or condensate in the center. And simultaneously, we do see a slight decrease in the DNA intensity, meaning that protein is also binding elsewhere. So sort of 2 modes of binding it is displaying. Here is the video that shows. The one mode is that it binds, condensates at the certain locations on the DNA. Other one is coating along the length of the DNA. You can also see in video that it condensates at specific locations on the DNA, and also it coats, or initially it starts to coat and then forms condensates at some locations, and you also have those coating elsewhere. Right, to understand the specific position, I'll skip this one. Yeah, will be done in a couple of minutes. So to understand where exactly the specific locations on the DNA that condensate are forming, so we prepared a DNA molecule with a specifically slightly higher AT content in the center, 3 kb AT rich region. You see there is a small bump in the AT content in that DNA. Of course, if you look at along the length, there are regions of 10 base. This is the 10 base window. What you're seeing, the AT content in the 10 base window. You see there are many regions which has a higher AT content. Protein can specifically bind there. But we also introduce a slightly longer AT content in the center. You could see that immediately when we introduce protein onto this one, at the position where we had this 3 kb AT rich sequence, there only the condensates are forming. Elsewhere, it's just coating. So the two modes of interactions are probably a little bit more clear here. That one position where it can form condensate or co-condensate at the position where you have high affinity region. Other regions it binds and unbinds much faster. And we could quantify a large number of molecules, always, almost all the time. We see the position where we have high AT content, that's where it binds. Right, from there we develop a model, or we provide a model for how this protein may work. We think that to be able to control large number of genes in the bacteria, it pulls those genes of which carries higher AT content into a one condensate, so that those genes could be controlled. And we have been working with Sandeep is there in the audience. He'll talk about this theoretical framework of this one in a couple of days. So we are looking at how these protein-protein interactions and protein-DNA interactions together will facilitate such a phase-separated co-condensation by physical simulation. So he will talk about that. But yeah, this is a snapshot of similar approach. A similar thing could emerge when you have high affinity and low affinity regions in the DNA. That probably will explain how it can compact the genome. Also, it can regulate large number of genes. So this is the last slide for my data. But of course, I want to thank all the people I've been working with, and also the funding agents that are contributing to our work. This work, the first part I was as a PhD and postdoc I did in KSD and Elio's lab in TU Delft. Now he moved to USA. The second part, the LSR2 work was done by very different people, like Tejas. He just moved to Case Lab. He was a UG student in the lab. And also Prakshi, she's there in the audience. She and Tejas teamed up to do that work. And that was with the help of Rohit. He was a PhD student in Sandeep's lab. And Sandeep will also talk about this thing. And I want to thank all of you for listening.
Thank you very much, Mahipal. So we have about 5-6 minutes for questions. Yes, please.
To the aggregation of this protein and that bind to the promoters and enhancers, right? Primarily, yes.
SMC proteins, you mean?
Yeah. Right. And these interactions are actually not static. They are kind of like dynamic, such that they come and go. So how do you explain this phase condensates in that context? Whether these condensates also form and disassemble, how do you explain
So
this?
yeah, the two different things. The first part, when I was talking about SMC proteins, they form the loops. They introduce the loops. The condensates also, we are saying that they also introduce loops in the genome somewhere or other, but these are more dynamic, of course. I mean, both are dynamic. There is no question about the dynamics of each of those components. But in the case of phase separation, of course, you have a large number of protein molecules acting at one place. So probably they are more efficient in organizing or pulling the component or DNA into one condensate than an individual protein when it comes to loop excretion, probably. But yeah, when it comes to actually reality, I'm not sure what would be the way there. Yeah.
It's a nice talk. So I had a question that where you showed that where you place 3 kb long stretch of rich DNA, there it's starting forming condensates, but in a genome manner where it's in an in vivo condition, so there you don't see this long AT rich things. Then how could you explain that inside genome?
Yeah. So in the case of MTB, there are horizontally acquired genes which are typically higher AT content than MTB genome itself. and those genes are thought to be regulated by LSR2. And then so we are doing analysis of the stretches of 80 rich genes or 80 rich segments on the genome. We see around 500 to 1 kb length fragments are there. So probably that is good enough for phase separation to happen. So we will check the different length 80 content DNA molecules.
Hi, thank you. Very interesting talk. It's sort of a related question, but with the other part of the talk where you talk about plectin pinning. How big of these sequences, curvature, intrinsic curvature sequences, do you need to pin plectinims?
Right, so we think around 100 bases is good enough if you have a really curved sequence. In the calculations, we took into account 70 base window as the segment, individual segments. So if you have a strongly curved sequence of 70 to 100 bases, that's good
You
enough.
talked about being those sequences being there in near gene promoters, and so those are like these 70 base pair. Are
Right, yeah. So they're right there, 100 base pairs at the start of the promoter. They are of roughly 100 bases or so in length. There may be some sort of genic elements that what we think promoter enhancers sort of, or maybe, I don't know, suppressors.
There's a question there from Dimple.
Right. So yeah, I was just answering to Indra, saying that the protein is evolved to act on the horizontally acquired genes in the bacteria, in MTB case, and those are the genes which are implicated in pathology. So those genes which are acquired horizontally probably are regulated by this protein. Yeah, they are AT rich. So MTB in general has a very high GC content. Most genes that are acquired are much lower GC content or higher AT content than the MTB genome itself. So they are there, those genes which carry stretches of long stretches of AT, higher AT content.
So just to follow, this is specific to this MTB case. It's not a generic.
So obviously LSR2 is there in MTB. The functional ortholog of the protein is HNS in E. coli, for example. People think HNS is also AT-rich binder. So we don't know exactly whether that HNS will also display similar mechanism. Yeah, we don't know.
Yeah, very interesting. So how do you see now the crosstalk between supercoiling and phase separation? Because you show that there is also sequence dependency here.
Right.
And phase separation is also would hide topological constraint potentially to the DNA. So
right. Other student in the lab, she will talk if you wait for a couple of hours, she will talk about some aspect of supercoiled DNA and DNA protein condensates. It's not really, we don't describe them as a condensate, but another nucleotide associated protein that has a higher preference for supercoiled DNA. In the case of LSR2, we tried to we didn't do really, look into it. But what we've seen that, with very small amount of data we have, somehow LSR2 do not care whether supercoil is there or not. It's very different result than what other student is going to present. Yeah, it's supercoil still, the plectin still diffuse while the condensate is there.
Last question, and then we'll.
Treating this tuberculosis?
Right. So people speculate. It's not that we are doing first time, right? People have been working on LSR2. They speculated that either you somehow express it higher amount or reduce it. Either ways, the bacteria do not really function. Yeah, I don't know. What are the ways to control or stop the protein from functioning? Or maybe if we can inhibit the dimerization itself also. Yeah, there are ways, I guess, but we are not really looking into those mechanisms. Maybe our interest is mostly to look into the fundamental aspect of how these proteins function, but could be interesting to look into it, yeah.